In this video, we will discuss about the effect of DiaBZl on T cell receptors, engineered T cells. So, the T cells that are engineered with this receptors, that is 1G4HA-T, high affinity T cell receptors, and its wild type version, that is 1G4 wild type, and those T cells has a high sensitivity towards the tumor cells where antigens are expressed using this gene, that is NY-ESO1. In the previous video we have understood or established this cytotoxicity effect of the engineered T cells against the tumor cells with those antigens that are expressed by these genes, NY-ESO genes. In order to further validate their hypothesis, what it did is they knocked down this NY-ESO gene from the tumor cells, and the schematic is shown in this panel A, where they have targeted three different guide RNAs, and in those guide RNAs they have used this PO gene, and that they call as NY-ESO PURO genes. And since the guide RNA of this NY-ESO1 has been disrupted, it will not be able to translate this functional NY-ESO gene tumor cells, and these things are validated using the Western blot in the panel B, where we can see that in the knockdown of this NY-ESO gene we do not see the presence of this NY-ESO, whereas in the control case where NY-ESO is present, we can see the presence of ESO. And when they check the cells cytotoxicity at different concentration of effector cells and the T cell target cells, what they find out is that in the case where NY-ESO has been knocked down, the cytotoxicity has been reduced. That can be seen in when the effector cells and the target cells are in the ratio 1:4, and when the effector cells and target cells are in the 1:2. And we can see that in the case of wild type, since the antigens are expressed because of the presence of this gene, the cytotoxicity is higher with increase in the effector cells, whereas it is almost negligible in both the cases when the NY-ESO has been knocked down, indicating that it's the antigens that are being targeted by these T cell receptors, not some other phenomena is going on. And when they did their experiment, and they further did their experiment with different concentration of the DiaBZl. So in this case, what they did is they treat the T cells and engineered T cells and these tumor cells, that is ME1-152 tumor cells with DiaBZls, to check whether the DiaBZl has some effect or it can enhance the cytotoxicity of the engineered T cells or not. And what they have find out is that compared to the case where the cells are not treated with DiaBZls, and they have find out is that in both the cases, that is when the T cells are treated with DiaBZls or when the tumor cells are treated with DiaBZls, they see the increased cytotoxicity compared to the case where cells are not treated with DiaBZls. And these things they have observed for the different ratio of the effector cells and the target cells, and they find out is that as the number of as the amount of or the ratio of effector cells increases compared to the target cells, they see that increase in the cytotoxicity. That can be seen in the panel D, E, and F. And one thing that they find out is that after after a certain threshold, the increase in DiaBZl will cause reduce in the cytotoxicity. That can be seen here, that is at 10 microgram and at 10 microgram in both in the case of this treated MEL 526 cells and treated this engineered T cell receptors for both the cases, that is with increase in the DiaBZl concentrations about 10 microgram, and but at the same time with increase in the effector cells at the same conditions, we see that there is a reduction in the cells cytotoxicity compared to the case where DiaBZl is used as 1 microgram in all the three cases. So from this experiment, they are able to find out the optimum conditions for DiaBZls to be used to increase the cells cytotoxicity, to increase the engineered T cells cytotoxicity against the tumor cells, that is 1 microgram. And when the effector cells and target cells are used in 1:1, and DiaBZl treatment is done using 1 microgram, then in this case we will see the higher cell cytotoxicity, tumor cell cytotoxicity against the T cell cytotoxicity against the tumor cells. So this is what they have find out. They not only validate their previous hypothesis but also find out the conditions at which they get the optimum cytotoxicity. That is, when the T cells are treated with engineered T cells are treated with DiaBZls at 1 microgram concentration. If you need to understand more about their experiments and how they have knocked down this NY-ESO gene, and what are the other reagents that they have used for their studies, and how they have validated why they have not used 1G4 wild type, you can go through the reference that has been provided in the description. However, I have already mentioned this reason why they have used IG4HA compared to the wild type in the previous video, where they have find out that IG4HA has more cellotoxicity compared to the IG4 wild type in their respective conditions, or in a particular conditions, not respective, in a particular conditions. So that's all for this video. Thank you for your time.